So, Chapter 2, Part 2, we're talking about isolation and the techniques that we can use to do that. So, streaking a plate. So, when you streak a plate, basically you are using what is called an inoculation loop. You can see that there's a stick with a little loop at the end. Okay. You are going to dip that into your bacteria, and on the plate, you're going to put a dot. So you have one drop of this, and it's going onto a plate. When you streak, you're basically trying to spread it out. And every time you spread it, you're trying to make it less and less bacteria. So you put your little dot, you fire your loop, killing any extra bacteria that are there. That means whatever was in that droplet is all you have. That's the highest number of bacteria that you have. So you're going to fire that loop, killing everything on it. And then you're going to go back and you are going to spread that droplet. Spread it about 5 streaks, 4 streaks. Then you're going to fire your loop again. Now remember, I had them all in one spot and then I spread them out. So there are fewer now in that spread section than in that initial dot. So I fire my loop again, kill everything. From where I spread, I'm going to spread again. But remember, I just fired my loop and I killed everything. So as I'm spreading again, this area here has an even lower number than my drop. So I'm spreading fewer. I fire my loop again and I spread again. As I continue to do this, every time that I spread, fewer and fewer bacteria are there to actually spread out, meaning that I have individual bacteria now on the plate that can now make individual colonies for me. So I know I'm isolating one specific bacteria. Okay, now loop dilution. So in a loop dilution, you are basically diluting your sample. So you take your loop, you dip it into your first vial that has stuff growing in it, and you put it into a fresh new vial. So you dip that little circle, kind of like bubbles from when you were little, has a film of bacteria in the middle. So when you dip it into the next vial that has nothing in it, you're decreasing the number. You take this one. Now, mind you, just did this, just mixed it and did everything else. I'm going to take the loop, I'm going to flame it, sterilize it, dip it in here that has a smaller number, and put it into a new one, which should make it an even smaller number. Every time that I do that, going from A to B, B to C, C to D, I'm leaving fewer and fewer bacteria in the liquid. Now, once I've done that, I'm going to take samples from each of these dilutions and I'm going to put them on a plate. As you dilute more and more and more, you get fewer and fewer bacteria in the fluid, meaning you're able to isolate colonies. Now, this is a lot more work than this is, which is why most of the time people do this. They do the streak plate because it's a lot faster and it's a lot easier. Now, spread plate. Depending on what your ultimate goal is, you may take a sample of this fluid, put it on a plate, and then you get something called a spreader bar. Okay, you can see it right there. Some people call it a hockey stick because it looks like a hockey stick. But you put that fluid on there and then you spin the plate. That hockey stick, like a squeegee, is basically going to spread that fluid around the surface. Now, again, If you're looking for something with very, very isolated colonies like this doesn't work very well because you're putting a high number and then you're spreading it all over the place. So, depending on what you want for your end result, this may not be what you want. You may want something like this or this. The technique you decide to use aligns with what your goal is. What is your goal? How do you want isolated colonies? Do you just want as many colonies as possible? What are you looking to attain? Okay, so inspection. Inspection. You've got cultures that you are observing macroscopically. So micro meant small, macro means big. In other words, I'm going to look at the plate and with my eyes get a description of what I'm seeing. If we go back, what are these? Well, let's say I've got a red colony. It's circular. I've got I don't know, off-white colony. It's circular. If I look at it, is it popped above the auger or is it concave like it dug into the auger? All of these are macroscopic, big, with my naked eye, things that I can observe about what's growing on that plate. So, looking at the colony, morphology, what color is it? What texture is it? Does it look super shiny or does it look dry? How big is it? If we go back and look, these off-white are always really big. Red can be pretty big, but the yellows always seem to be very small in size. That's another clue that you're dealing with something different. Okay, so that was the wrong computer. The specimens can then be observed microscopically. You have techniques for making slides so that you can look at them under a microscope and actually look at the cells themselves. What is their shape? Are they big? Are they small? Are they mobile? Do they move around? Um, Depending on which tests you use, you'll be able to tell those types of things. So, we have one bacteria called Bacillus megaterium. Mega means big. This is a hot dog shaped bacteria that is huge compared to everything else that we see. So it's called B mega or Bacillus megaterium. Um, as far as the motility, we have a method called the hanging drop technique. And in that technique, the bacteria that you're working with stay live. They're still living bacteria. And you look in the scope and you can see them swimming. Some aren't mobile. So if they're not modal or mobile, then you won't see any movement because they're not. Now, identification. You do tests to try and identify the organism. Different types of stain on slides stain different types of cells. You've got techniques that separate certain groups of cells from others. So you can use cell staining to kind of narrow your field. I know I've got a gram positive, not a gram negative, because it stained this color. Biochemical tests. Does it have hydrogenase? Well, if I put hydrogen peroxide on it, peroxidase. If I put hydrogen peroxide on it and it doesn't have peroxidase, there will be no bubbles. However, if I put hydrogen peroxide on it and it bubbles, that means that specific type of bacteria has peroxidase in it and can break down hydrogen peroxide into a gas and water. So you see bubbles. That is another identifying feature for specific bacteria. Some of them can't do it, some of them can. So it's again a way to kind of narrow into your identification. Immunologic tests. Do certain antibiotics work on it and others don't. Usually, when you get an antibiotic, it is because you have a specific type of illness. Antibiotics only work on specific targets. So if you've got a bacteria that antibiotic doesn't work on, you're not going to get that antibiotic. DNA analysis. Now, this is getting really complex. You can isolate the DNA from the bacteria. You can get it sequenced, and you can look and analyze what genes are present and everything else. The results from the tests are analyzed to find out what microbe we're working with. Now, most of the time, you don't have to come to that. You don't have to use immunologic testing and DNA analysis, at least in our lab. In our lab, that is really not necessary. But if you were actually working in a real lab where you are trying to isolate and figure out what things are, you may go this far. Okay, so a microbiologist's best friend is the microscope. Give me a second. Okay, so we mentioned this in chapter 1. Microbes are microscopic. They're too tiny to see with the naked eye. With that in mind, we need an instrument that will increase their size enough that we can see them. The microscope is the instrument that does that for us. We primarily use the light microscope for most of our microbial applications. But there are other types of microscopes: electron microscopes, confocal microscopes, fluorescent microscopes, phase contrast microscopes. There are a variety of them, but the kind of standard for most colleges anyway is the light microscope. The main goal of microscopy is basically twofold. I don't just want to magnify my specimen and make it bigger, which is basically what that means. I'm making it bigger. I also want to keep good resolution. Resolution is the ability to distinguish 2 points from each other. It basically just determines how clearly we see something. So I don't just want to make it bigger and blurry. I want to make it bigger and crisp. I want the image to be crisp enough that I can see the fine details. So if you have a good microscope, both things should be taking place. Magnification and resolution should both be good. So, clinical focus. When it comes to collecting patient samples, it's critical. A critical step in that process is being able to isolate, grow, and identify the specimen. Basically, you're looking to figure out what this person has. Improperly collected samples can result in contamination of the specimen. You don't want contamination because then you can incorrectly identify what's making this person ill. Contamination generally comes from the normal biota, the normal bacteria, the normal microbes on the patient's body, or from contaminants from the person collecting that sample. Contamination can lead to failure to accurately identify. Like I said, if you don't realize that you got this from somewhere else, you're not going to identify it correctly. That means ultimately that you cause problems because now they're treating the patient for something that isn't the right thing. Identification is key to being able to come up with a treatment plan. And if you don't identify correctly, that treatment plan goes out the window. So you have to identify correctly. Contamination just basically can ruin it. When collecting patient samples, be sure to follow all standard protocols, such as cleaning a wound prior to collection, ensuring the patients follow the clean catch urine sample procedures. If If you've ever had a UTI, you don't go in and pee in a cup and that's it. Usually they hand you something that looks like a wet wipe. That wet wipe is to sterilize the outer area around the urethra so that when you urinate into the cup, the only bacteria that they should find is the bacteria within the urine that came out of your body. It's not coming from around that space. Um, Ultimately, when you collect, it's kind of like when they take blood. They don't just stick a needle in your arm. They will sterilize the area with an alcohol pad first and then kind of let that dry a little and then collect their sample. One of the reasons they do that is because they don't know what kind of bacteria are on your arm. If there are bacteria on your arm, kind of double danger. I can actually use that needle to jab that bacteria into the inner part of your body, or I may contaminate the sample that I'm collecting. If, again, I want to identify something specific that's happening within your body, I can't do that. So, when it comes to collecting samples, you always make sure to follow the proper procedure because if you don't, you're messing up your patient.